All right. Dr. Jonathan Miner here to give you another update on RVCL. It's 10 o'clock on Saturday night, had a long day. It's been a while since I gave an update. I want to try to do this more frequently. I apologize, it's been a little while. A lot of news. I've been very busy and some very good progress. So I'm going to go ahead and share my screen and so you can see what we've been up to. So my first interactions with families with RVCL, as I look back over the last decade, include some of these kinds of comments. Uh, in reference to a scientist who was looking to develop treatments, I remember one family member mentioning that they liked that that person was focused on treatments. These are things that I heard and have been uh, that I internalized and that have affected my thinking. Another comment is the more people involved in RVCL research, the better, which is something that I really bought into and is a big part of what we've been doing here at the RVCL Research Center at Penn. I'll show you in a moment. We recently discovered the mechanism of the disease, and it's been this massive international collaboration, 4 continents, 40 different authors on our paper. And And then there are people who've said things to me like this, like, you're the person that's going to save my life or my kids' lives. And, you can imagine the pressure that I feel with that. But at the same time, I am committed to the possibility of achieving that. And I think we will achieve that. I can't predict the timing, but I can say that I'm doing everything I can do to make it happen fast. I took this seriously, this idea of more people being involved. I took the idea of openness and working with other people very seriously. We enlisted help from all around the world. These are our colleagues in Japan. They have patients with RVCL as well. These are incredible, wonderful people. Up front here is Taisuke Kato, an incredible scientist, fundamental to the discovery that RVCL is a DNA damage syndrome, or a syndrome where chromosomes become injured. And here in back is Osamu Onodera. He's the head of the Brain Research Institute in Niigata, Japan, on the west coast of Japan. And when you see him standing in the back there, this actually epitomizes his approach. He's in back, letting the young people lead the way, not trying to take credit for all the work, but instead trying to lift other people up. These are wonderful people who are committed to developing a treatment for RVCL. And our interactions with them and our openness and sharing has been transformative. These are folks in Australia, all incredible people. Tristan Hardy, for example, is a clinical geneticist who reached out to me one day. Another key to the discovery that RVCL is a DNA damage syndrome. Toby Coates, a kidney doctor, another great collaborator. He was just here at Penn weeks ago. Amazing story. He's taking care of families with RVCL, parents and kids, and many others. There's another Dr. Hardy, who's a neurologist in Sydney, Australia, that has worked really well with us. All of these people have been wonderful. Our collaborators in Scotland are amazing. This is the Royal College of Physicians, where I presented on RVCL in Edinburgh, Scotland. Uh, they have done some remarkable work as well. Also found evidence that RVCL is a DNA damage syndrome. Incredible collaborators in Paris, in France, Yasmine Belcade and Nicolas Manel. Nicolas Manel has visited us twice here in Philadelphia from Paris. A good friend and a great scientist. These interactions are essential to solving RVCL. This cannot be about credit and someone taking credit for what's going on in the disease. or saying, I'm the solution. The solution is all of us working together, patients, scientists, doctors, sharing openly. Michaela Gack, an amazing collaborator from Chicago and University of Chicago, and then later in Florida. Max Artemoff, who's my good friend, a computational scientist at WashU, who and great bioinformaticist, has helped us a lot. And then we've been offered help by our neighbors at Penn. So part of why the culture at Penn has been so amazing is our neighbors just across the hallway come over and say, hey, have you thought about this for RVCL or that? Rahul was the person who got me on track for gene therapy for this disease. More on that later, but I should say that we now have confirmed that we can fix the disease-causing mutations in animals with an intravenous injection. Now, it's not going to fix the mutation in every single cell in the body, but in a significant percentage of them, this could be a path toward cure as we optimize this. John Wary, another amazing immunologist here at Penn who has helped us tremendously. He calls me and offers support. It's been very touching the way people have bought into our goals and to facilitating them here. This office where I'm sitting used to be Drew Weissman's office. He won the Nobel Prize. And he's been helping us as well using a gene editor that we developed to treat RVCL. And he's made an RNA version of that, which we're hoping to be able to give to patients repeatedly to fix their mutations. The interim president of the university knows about RVCL, Larry Jameson. He's met with patients who have RVCL. And again, incredibly supportive of our efforts and understanding of what we're trying to achieve. These are the Coltons, Judith and Stuart Colton. They donated $60 million to Penn, and $300,000 of that has gone to developing therapies for RVCL. He came up to me after one of my talks, and I told him how much the patients appreciated their support. And it was very touching for him and also for me. They understand how bad this disease is, and they want to help. And being here has allowed us to enlist help from, as you can see, people from all around the world, but also from people like the Coltons. We're moving to this new research building next year. We're going to be at the Colton Center for Autoimmunity here on the 15th floor. We're very excited for that. It's going to be nice space to try to move things even faster. Now I want to talk about the fruits of hard work and support. This is the eye of a fruit fly. It's actually two fruit flies that have been spliced together. Someone in my lab said that this looks like a strawberry, and I can see the resemblance. The lower half is from a fruit fly that has the RVCL causing mutation in TREX1. And you might think that we're too different from a fruit fly to learn much from this, but actually there's enough similarities at the DNA level that we can learn a lot. And so these fruit flies were made by our collaborators in Japan, and it really advanced our work tremendously. So here the word fruit has multiple meanings. The DNA damage theory of aging. Would you believe it if I told you that these two mice, even though they look quite different from each other, the one on the right looks older, these two mice are siblings. They're essentially identical twins except for one gene that is causing injuries to chromosomes. There is a theory of aging called the DNA damage theory of aging, that over the course of life, we accumulate damage to DNA in all of our cells, chromosome breakages. That these breakages cause cells to age or to die. And we discovered, along with our collaborators in Japan and from all around the world, that this phenomenon is central to what is happening in RVCL, in our patients. Understanding this, in our view, is the key to developing treatments. So we recently published a huge paper that represents many years of work in Nature Communications, you can see there are 40 authors from 4 continents, all incredible people who have given so much of their lives in order to finally understand the mechanism underlying this disease. You can see all the locations from all around the country and the world. We got the first large NIH grant on RVCL that includes avenues to a therapy. We've had support from the Colton Center here. We discovered that women with RVCL are at high risk of early onset breast cancer before age 45 or 50. That's something, if you have questions about, please reach out to me. I'll be happy to update you about what we found. But it's very similar to if a patient has a BRCA variant. I'm going to do another, that's a genetic variant. I'm going to do another video on that shortly. The key thing is we discovered this is a syndrome of DNA damage. I've given countless talks with feedback from many thousands of scientists from all around the world. And that feedback, that openness has been essential to us making progress. Now we have a gene therapy that can fully correct the most common mutation in mice. There's still a lot of work to do to make that available for humans. We have a protective small molecule drug that could be taken as a pill. There's still work to do on that as well, but that was developed at the University of Michigan. We have unique tools that I generated immediately upon arrival at Penn. As soon as I came here, week 1, I started making new animal models with human mutations in them that allow us to test these therapies. These are tools that we did not have before. They're different from anything that existed previously, and they were designed specifically with therapy in mind. We have gene therapies now that we can deliver either by modifying a virus. We take out all the virus parts and we put in the gene therapy parts. Viruses are really good at delivering DNA into a cell, which can then edit the genome because of the way we've engineered it. It has basically a genome positioning system, GPS, that will go right to the mutant TREX1 gene and fix it. Now, it's not 100% of the time. We have to work on the efficiency, but it's pretty good. And then we have other ways of delivering nucleic acids like RNA and DNA to cells. With our collaborators, we've screened 7 billion compounds and narrowed it down to hundreds. We have lead compounds, but there are others that we're working on right now. We're enlisting help from all around the world from chemists making these. The chemists have to synthesize the compounds once we've identified the ones that interact with TREX1. We can take pictures of the TREX1 protein and how the drug interacts with it. This is helping us make better and better drugs that could be taken as a pill. We have to screen them as they come in, so our collaborators make them and send them to us. Now that we know that it's a DNA damage syndrome, it's really easy for us to test them. So that's going faster and faster. We need a lot more laboratory work before we're ready for a trial, though. We're moving as fast as we can. Our team is exhausted and overwhelmed. I'm here way past 10 at night. I'm not the only one. We want to thank all of you for supporting our work. I hope that you all know that for us, for me, but also for others here at Penn and for others around the world, like our collaborators in Japan, patients are what matter most. That's why we're doing this. This isn't about glory. This is about the people we care about and saving their lives. That is our goal. That is our focus. It's also not about how smart we are. I actually think that most good scientists, the best scientists, are not necessarily geniuses. My view is that the best scientists are good communicators who are open and who work really, really hard. So I like to say genius is defined by the work that you do. We are working like crazy to get this fixed. So what helps us go faster? Well, the patient and family support, the donor support, the grants, those are all making a difference. We need hands at the bench in the lab working on this. Bench scientists, chemists, people synthesizing and testing compounds. We've been recruiting really well. I've just added 2 new people over the last couple months. Things are looking up. Things are looking up. Gene therapy, it's effective in mice. 23% of cells within the liver with one dose. This is kind of complicated to look at, but this is a diagram of the TREX1 protein, and you can see that this is the abnormal TREX1 protein here, this black band. And we're able to, when we give the drug, it's a plus here, we can convert it to the full-length normal TREX1 protein, which is quite remarkable. And we do it with good efficiency. And we've been able to do it in animals. That's the key step. Doing it in animals, making sure it's safe, making it more efficient. This is a path toward a cure. I'm not saying that it's going to happen next year or the year after that, but it might. We're trying to make it happen as fast as we can. We definitely don't want to delay. We want to go fast, but we also want to make sure that we do this safely and responsibly. It's important to do it the right way. But I'm committed to keeping you updated. We certainly aren't going to hold anything back. It breaks my heart every time a patient, their disease course worsens or that they become debilitated or they pass away. My heart goes out to every family member and to all the patients who are suffering with this. And every time I tell a patient they have a mutation, I tell them there's still time. But when I go to the lab, talk to the people working at the bench, I say, remember, we are in a race against time, not against other people. We want to work with other people, but a race against time to save these people's lives. That is my commitment to all of you, that we view it that way and are working as if the patients are our own families. So the future, clinical trials. Clinical trials, I talked about a trial at the beginning, but I think the important thing is understanding that we're talking about clinical trials that really meaningfully slow down and stop the disease, personalized, designed for RVCL, molecules that target TREX1, custom designed for our patients. We have great expertise at Penn in clinical trials, paths toward FDA approval. Penn is one of the leading institutions as far as getting drugs FDA approved. We have to improve these small molecule drugs so they're going to be taken as pills. There's still a lot of work to do to make them stay in the body longer, to make sure they're safe. As I mentioned, we have the gene therapy working in the animals, but we have to optimize it further. There's safety testing that has to happen. We need a small army of scientists, bench scientists. We need hands to get this done. And so, I just want to close my video by saying thank you to all of you. Thank you for your trust. Thank you for working with me. Thank you for reaching out. And please know that I am always here. It doesn't matter what time of day or night it is. I'm on call 24/7 for you. You never have to worry about bothering me. I'll make myself available. And if I ever don't respond, it's not intentional. I get like 800 emails a day nowadays. Just send me another one and I'll get back to you. Don't be afraid to reach out. Thank you and have a good evening.